No, I'm actually impressed this many people showed up.
Welcome everybody to this morning's event for the Cambridge Science Festival here, and thank you for coming through the snow. Clearly a few people decided it was all far too tough. I'm glad they don't live in Canada or Sweden or anywhere else like that. But it's great to have you all here. I'm Julian Puppet and I run the Intellectual Forum here at Jesus College. If you've never been to Jesus before, you are very welcome. It was originally a 12th century nunnery, then became a college in 1496, so we have a very long historic track record. But we also do try to move with the times. You may have spotted this room wasn't built in the 1140s. It was in fact only opened last October. An intellectual forum which I run is aimed at trying to get people to think and talk about anything that's interesting and worthwhile. So we've done all sorts of things about international law, we've had Jimmy Chu come here, we have some amazing events on all sorts of subjects. So I hope you'll stay in touch, come along to some of our other events. This morning we have what should be a really fascinating talk from Jim Ajioka. So Jim is a fellow here, looking after some of the medics, he's a pathologist. But today he's going to talk about quite an interesting application of that, of bioengineering for the fashion industry. So it should be really interesting. Jim will talk for however long he wants to. We'll then have a chance for questions. There is a microphone, so my deputy Sarah has the microphone. So when we get on to questions, if you could hold the microphone close to your mouth, that would be very helpful, and wait for it to come. The other thing to say is we are filming today. If you would like not to be caught in any of that, please let us know. We have a few cameras around. And in the event of a fire, which we are obviously not expecting, please go out the door there and go back into the court behind us. Not onto the road, where the fire engines will be arriving, but onto the court, where you'll be much safer. And we'll give you further instructions at the time. So, thank you very much. Jim, it's great to have you here. Over to you.
Okay, thanks. So this is not my, is this working? Is this microphone working? Oh yeah, it is, okay. So this is sort of a hobby that has been going on in the lab for about the past 2 years, and so this is not what I normally talk about, so I'm a little bit nervous in doing this. Okay, so like every other project, there is motivation as to why you do something. Okay, water, water everywhere, nor a drop to drink. This is a quote from the Rhyme of the Ancient Mariner, and a poem written by Coleridge, who was a student here back in the 1700s. And this poem is about basically how nature has been disrupted, and there's redemption associated with that. In Nepal, where the story starts, it's one of the most water-rich countries on Earth. Okay, there's lots and lots of water. But ironically, a lot of the people, in fact, a good proportion of the people that live there, do not have access to clean water. So the story starts because we went to Nepal to look at ways of making an arsenic biosensor. What we were trying to do is to engineer bacteria that would respond to arsenic at toxic levels and then detect and report that back to the user, so you could test your water and see if it was at toxic levels of arsenic. Arsenic contamination in drinking water affects 100 million people across the region. This includes West Bengal, southern Nepal, and most of Bangladesh. So there's a lot of people that are affected by this problem. So why do we have a problem with arsenic? This is a technological problem, essentially. About 20 to 30 years ago, most of the governments across the region decided to drill shallow tube wells to provide pathogen-free water to people in villages. Well, unfortunately, a high proportion of those shallow tube wells that were drilled hit aquifers that are laden with arsenic. Arsenic is one of the most common elements on Earth. And so the reason why we decided to use a biological approach is because most organisms have to detect and detoxify arsenic specifically because of that problem. And so we went to the villages to just basically see what was going on and talk to people. This is a typical set of sort of skin lesions on people that have had chronic exposure to arsenic. This is indicative of the level of exposure that the people in the village have, and also across the population, the morbidity and mortality associated with this. So we're looking at things like multiple organ failures and cancer. Okay, so we had partners in Nepal. This man here is Dr. Suman Shakaya. He is the director, or was the director of an NGO called ENFO, and they work on water quality of various sorts across Nepal. And so he took us to some of the villages that they've been working with in terms of education for arsenic exposure. And he, with his translator, allowed us to ask questions to people in the villages. And so since we were trying to make an arsenic detector, one of the things we wanted to know is what they would actually like to see in the detector itself. And they said, well, we need, if it's going to be something that's going to be useful for us, we need to have it a very clear kind of stoplight type arrangement, and we need something that was very visual, a color change that will tell us when our water is drinkable or not. So this is what we ended up doing. We made an arsenic detector. We built several different strains of bacteria that would respond to arsenic at different levels, and made it into a stoplight type arrangement. And as you can see, at 50 parts per billion, it's responding in the way that we would like, we've engineered it to do. And this is with water that was derived from a well in the Terai in the southern part of Nepal. And when we tested the water itself with spectroscopy, it turned out to be just above 50 parts per billion arsenic. And this is a level that's barely acceptable for drinking. The WHO level is really 10 parts per billion. So this is clearly in the dangerous range of drinkable water. Okay, but then we went back to Kathmandu, and we were working with the guys at ENFO. There's still places that in urbanized areas that have real problems with water access, but for very different reasons. This is one of the aqueducts in Kathmandu, and what you begin to realize is they're heavily polluted. And they're heavily polluted in ways that there's a major concern, because the factories that line the aqueducts in the river are things like tanning factories and textile dyeing factories. And these basically dump all of their waste into these aqueducts, making the water completely undrinkable. And unfortunately, a lot of the chemicals that they dump into the aqueducts and rivers are not retrievable. And so once they're in the water, they're there forever. And so the water is stagnant, and it's really not drinkable. Even the feral dogs in the city won't drink it. So this is the real problem that they're concerned with in urbanized areas. What? It's just garbage. It's just plastic bags, bottles, all sorts of stuff. Okay, but it's just indicative of what's going on in, yeah. So why is this? Well, most emerging economies turn to the textile industry as their first mode of business, because they become instantly globally competitive, because they can make things that are very reduced cost. Unfortunately, part of that cost is the lack of regulation, and as a consequence, a lot of the regulation for things like textile dyeing are basically non-existent in Nepal, so they can just dump the waste directly into the rivers, and that is why we have this problem. However, it turns out it's not just a problem in Nepal, but it's a problem globally. This is a fairly iconic image of Xintang, China, where there's a denim factory that's just above about where the ceiling is, that is dumping indigo waste into the river, and you can see how polluting that can be. And so this is not something that is just a Nepalese issue, but something that's global. Okay, so we're going to switch gears here now. So you kind of see why we started this project in the first place. We understand that there are real issues with the textile dyeing industry in places like Nepal, Bangladesh, and other emerging economies. Engineering biology, this is what we do in the lab. Okay, the key realization that biology is a manufacturing capability is a quote from Tom Knight. Tom Knight is a really smart guy. He used to work at MIT. In the artificial intelligence lab. He is one of the people that invented the internet. But about 30 years ago, he decided that silicon had seen its day, and decided that biology was where the next big manufacturing capabilities was going to happen. So he started learning biology. And Tom started a company called Ginkgo Bioworks, and if you ever look that up on the internet, you'll see that it's a very big company, and it's actually something that's probably the best company in this space right now. Bacterial cells are factories. This is something that most of us had realized, but didn't really take on board what we could do with it. So if you look at bacterial cells, they have a control system, they have environmental sensors, energy producing capabilities, they have communication systems, even motility. But importantly, they have high production capacity. If you want to make certain kinds of products, you can get them upwards of 10 to 30% of the total mass of the cell. So we can make those things produce a lot of stuff. Okay, so Tom and his colleagues at MIT started a competition at MIT called the International Genetically Engineered Machines Competition. And here in Cambridge, we were the first international team. In fact, that's why the I is there. And this is something that a couple of colleagues of mine, Jim Hasloff and Goss Micklem, we supervised teams of undergraduates during the summer to make genetic circuits that would do something cool. And in 2009, we had students from the Royal College of Art working with us. And they, they're artists, and so they wanted to see something visual. So we decided that we would make a whole bunch of pigments. And obviously you can see that it worked, and the project was called E-Chromi, and we actually won the grand prize that year for this project. Okay, before I go on, we think we need to quickly review the central dogma of molecular biology. DNA makes RNA makes protein. Okay, this is the functional part that underlies all of life, and it allows us to be able to control what we do. Okay, so it's the control of this process that's very, very important in our ability to make cells become factories. So when Tom started out this idea of making genetically engineered machines, he realized that being an engineer that you need standard parts. Okay, so the standard parts are effectively things that are DNA encoded to control the making of proteins, but they're all encoded on the DNA. So you can separate all of this into what we call parts, or biological parts. So you want parts that will control RNA production, protein production, the protein code itself, and RNA termination. You know what things to know when to stop. Importantly also, for an engineer, you want to have modular parts. You want to be able to swap things in and out, and you want to be able to make different combinations of parts to make different kinds of machines. Okay, so modularity is a very important issue when it comes to this. And what is important is all of these parts are characterized to some extent, so you know how they're going to behave in the organism that you're putting them into. Okay, so if we look at the part function, some of the most important things are things like the control of RNA machinery. The control of RNA machinery can be encoded on the DNA itself, and it acts basically like a gatekeeper. So it will tell the machinery the correct position to assemble. It'll tell the machinery when to start and how much RNA to make, and that can all be encoded on the DNA. Oops, sorry. On the other end of our code here is that we need to control the RNA machinery to tell it when to stop. We can also encode the control of protein production. This won't be functional at the DNA level, but once the RNA is made, we can, in fact, control the protein production itself. So again, we can have a different gatekeeper type sequence that will control the protein machinery, tell it where to assemble and how much protein to make. The protein code itself will tell you the correct position to start for the machinery to start making the protein, which protein to make, and then where to stop. So collectively, we can take these parts and assemble them into a functional genetic circuit. Now, standard parts also need a standard way of assembling, so you can swap them in and out without having to change anything. And so Tom designed a system where you could have specific ends on them that would be compatible all the time with all of your parts. And so as a consequence, you can assemble the DNA parts and then put them into an organism. Okay, so now I've made this into putting them into a bacterial cell. And that piece of DNA will now be able to be read by the machinery inside the cell. So the RNA synthesis machinery is kind of like having a copying machine. It will go along the DNA. It will tell it where to assemble and make the RNA, and also where to stop. The protein synthesis machinery will behave more or less like a 3D printer. In this case, it will assemble at again, the gatekeeper site. It will make protein from that, but unlike the 3D printer, it doesn't have a moving print head, but all of the 3D functions that are encoded in the protein coding sequence itself, and so all the subunits, the amino acids that make up the protein, will determine how the thing folds into a 3D structure. So in this way, we can control all of the steps in making protein. Okay, so enzymes are proteins, and one of the great things about enzymes is that they can do things that at a very high rate, so they can lower the activation energy for any kind of chemical reaction. So in this case, we can take colorless compounds, combine them through an enzymatic reaction, and make colors. Okay, and that's what we do. Enzymes can come from anywhere in nature's palette. You can get them from effectively any organism on Earth. We all speak the same language, so we have a universal code, so we can take any enzyme from any organism and fit it into our bacterium and produce color. Okay, so like any other project, you have to have some level of inspiration as to what you're going to do or how you're going to do it. Okay, so everyone in this room, I'm sure, has seen this, okay? We've all accidentally left our tea towels on the side and they rot, okay? My wife yells at me when I do this, and she's sitting over here. Okay, so what you begin to realize is that once you put this in the washing machine, the color doesn't come out. You cannot get the mildew stain out. Well, guess what? That's microbes basically dyeing fabric, okay? So back in 2009, I took a piece of nylon and we had a pot of purple bacteria, and like every little kid, your first instinct is to draw your initials, okay? And so I did this, and this was fun, and so I fixed the bacteria on the piece of nylon, and then after the competition, I just threw it on my desk, and it sat there for 4 years. But one of the things I noticed is it didn't fade, okay? The color just never, and you can, you draw something with your biro and it fades almost instantaneously. So this was really kind of an amazing observation. So when my postdoc, Ori Arconi, came to work in the lab to do the arsenic project, I was showing him this, and then he kind of got slightly obsessed with trying to make this into a real process. Because if you look at both the tea towel and my scrawled initials, they're very spotty, they're not very even, and this is bad for dyeing fabric. You can't really do it. It's not a great process for that. But Orr figured out how to make it work, and that's kind of a result, okay? Once we do it, you can actually make it really, really even, okay? So how does the process work? So our process ingredients are first the recombinant organism that we make that can actually make the pigment itself. Our food is based on molasses. It's a sugar byproduct. And the thing that we use for the transfer media to make the bacteria stick to the fabric and then dye the fabric in the end is a biodiesel byproduct. So we're reusing a lot of stuff that's just byproducts from other processes. Okay, so we start by just taking the bacterium and the molasses medium and putting it into a fermenter. In our case, mostly it's just a flask that we shake at 37 degrees. Okay, so once you have a high density culture, you can take that culture, put your fabric in with the culture, put in the transfer media from the biodiesel product, and at 37 degrees and about 3 hours later, you can have a stained or dyed fabric. Okay, so then the fabric itself is covered with a genetically modified organism that contains the stain dye in it. Then we autoclave it just to kill everything. So we have to do this anyway in the lab because anytime you want to take stuff that's recombinant out of the lab, you have to kill it. Well, in this process, it kills the bacteria, but it also breaks open the cells. The cells dump everything onto their contents onto the fabric, and between the heat and that, it sticks to the fabric very well. And then you just have basically a stained, dyed piece of fabric with some residual bacterial stuff on it. So then you just put it in the washing machine and you got a clean piece of fabric. So it's not very complicated. And we actually tested the finished product to see if there's much in the way of residual bacterial stuff on it, and there isn't. It's actually quite clean. The test that we did is something that people use to test medical instruments for cleanliness, and while it wasn't quite close to being clean enough to use as a surgical instrument, it was close. There was more bacteria on the fabric our control fabric, which came from the shop, than it did after we washed it. Okay, so fashion in the circular economy. Let's talk about that for a minute. Okay, I'm going to start by talking about William Perkin. I don't know if any of you know who he is, but his 180th birthday was celebrated on Google last week. Okay, William Henry Perkin was a chemist back in the Victorian times in the mid-1800s, and he was trying to make a compound called quinine. Okay, quinine is something that's an anti-malarial drug, and back in the time when the empire was big in Africa and in India, a lot of people were dying from malaria, so quinine was quite an important compound to try to make, because unfortunately, I think the Portuguese or the French controlled the market, and that was not a good thing for this country. So unfortunately, he failed making quinine, but what he did do is make movine, which is a nice purple dye, and it was the first aniline dye that was ever made. And so Perkin can be kind of blamed for the petrochemical industry, because then a bunch of German companies like Bayer and BASF started out as dye houses, and then they became what they are today. But they all started out trying to make this sort of thing. Okay, so when people talk about circular economies, I couldn't really figure out what that meant, okay? So, but you can kind of understand what a linear economy is, okay? So the linear economy, whenever you look these things up on Google, you start out with something where they extract a non-renewable resource, they manufacture it, they distribute it, you consume it, and then whatever's left over goes into the landfill as waste, okay? The reason why this is linear is because it's a unidirectional thing where you start with something that's not renewable and you end up just making waste out of it. And that's probably not a very useful way of going about production. Okay, so when you look at the circular economy, it's actually many intersecting circles, not a single kind of thing. So it's based on the idea of reuse, reduce, and recycle. Okay, these are the three R's, different than the three R's when I was growing up, but the three R's that drive what we are striving to do. So even in the current industries for making dyes and dyeing textiles, they are trying to have circular economies within those different sectors. So currently, when you look at the textile dyeing industry, it starts with petrochemicals for the most part, so that you need oil-based things to make the dyes themselves. Mining is a problem because a lot of the clothes you're wearing have chromium in them as mordants to fix the dye to the fabric. Chromium is highly toxic, and it creates a lot of problems downstream. A lot of these industries, they can dump the stuff directly into the waterways, and they're, again, irretrievable compounds, and they're really toxic to life. Transportation costs are huge because when you start looking at the fabric dyeing industry, the dyes themselves are only produced in a few factories across the earth, and so they all have to be transported to the places where they need to go for dyeing the fabric itself. So the carbon footprint's high. The garments themselves, well, you can see what happens to garments, especially the ones that are based with chromium mordants, because those things are so toxic they can't recycle them, so they go straight into the landfill. So circular economy is what you're trying to do is to improve the RRR and make the circle sort of more impactful and then reduce the waste, okay? So again, you're trying to reduce the extraction, manufacture, distribution, consumption, and the waste itself. So what are we doing? So when we don't really extract anything, because one, we're just using basically renewable resources that are byproducts from other industries anyway. So at least we're hitting a couple of the R's when we do what we do for our inputs. We're more efficient because we can combine two processes into one, and therefore there's lower transportation costs. And ultimately, this can be pretty much localized anywhere on Earth so long as you have a sugar source. And the garments are certainly going to be more recyclable because there aren't things like chromium embedded on the fabric itself. So anything you have will be recyclable, or at least to be recyclable on the basis of what we put into it. Okay, so this is our company, Colorifics. We just started up. We're up in Norwich now. So let's look at what we do. Okay, so the byproducts that we use are from first molasses from the sugar refining industry. I don't know if any of you know much about sugar, but effectively you can recrystallize sugar out of a mash of, in this case, sugar beets. And you can do that several times, about 4 times, but after that it doesn't become economically viable to try to recrystallize anymore, and that's molasses, that's what's left. And so we went up to the sugar factory in Bury St. Edmunds, and they just laughed at us because we said, "Can we buy some molasses from you?" They said, "No, we'll just give you 10 liters because it's so cheap." And so they gave us 10 liters of it. And sugar beets are good because there's lots of other things in there besides sugar. So all of the things that you need to build cells like phosphorus and nitrogen get carried over in the process. And so in the mash, we still get nitrogen and phosphorus in there. Having said that, we've had to supplement our media a little bit because we haven't optimized the bacteria to use molasses, but we can do that in due course. The other thing we use in the transfer medium is biodiesel production. It's glycerol or glycerin. It's in everything that we eat, use as cosmetics or whatever, so it's really pretty benign, but there's a glut of it because of biodiesel production from vegetable oils. Okay, so again, it's one of these things that's really cheap at this point. And because they're both plant-derived products, they're renewable resources. Okay, so the bacteria that we use, they're domesticated lab strains. They're highly disabled. They can't compete in the environment, so there's really very little danger. They've been used for 60 years. There's very little danger of them. Actually, there's no evidence ever that they've gotten out of the lab and created any havoc at all. They're things that are used for, say, producing human insulin at this point, so they're well understood organisms. Because they're self-reproducing and because you can just grow them to any scale, they're really useful for trying to keep a circular economy in place, because you can take them, we can grow them in shake flasks like you see on the left, up to fermenters, 1,000-liter fermenters, and this is a well-known process. Okay, so compared to other conventional methods, we don't use any hazardous chemicals. We use about 10% of the water, 30% of the energy, and 1% of the waste. And if you look at the 3 pictures I've got up there, so the first is the growth media, so that's the molasses mix. It's pretty much diluted down to the point at which you can use it for growth media. We can put the bacteria in them, grow them to a dense culture. You can see that in the middle flask. It's so dense you can't see through it. But then after we put the fabric in and then we dye it, you can see what's left. So basically it all gets taken up, or pretty close to it. So we've got less than 1% waste. And because it combines 2 processes of making the color and depositing on the fabric, it really will lower the carbon footprint of anything you want to do. And this is just some examples of the sort of things we can do. So it turns out once you start looking into how things are done, people were really surprised that we could use the same process for both natural and synthetic fibers. Generally, they have to do very different processes to get them to dye. We can dye polyester without pressure, which is something I hadn't realized, but apparently when you dye polyester, you need to push it through at a high pressure, and we can dye polyester without doing that. Obviously, because we combine 2 processes, we can lower the transportation cost significantly. And at this point, we're the only new tech for dyeing fabric that has a viable scaling for industry, and that's really important. Nobody wants to see processes that won't be economically viable. In fact, talking to people in the industry, they're less impressed about the greenness of this and more impressed that we can actually do something that probably will be of lower or equal cost to current practice. Okay, so here are some of the brands that we're involved with at this point. And also we're in a couple of accelerator programs, BioStart and Fashion for Good. Dave Nally, who's a fellow here, I don't think he's here today, but his wife kind of works in the sector. And this is one of the things about being in Cambridge. So I gave a talk on the other stuff I do in the lab, and Dave's wife came and talked to me, and we started chatting about this business. He said, oh, here's a phone number for somebody that might be helpful. And so I called this guy up and said, oh, you can do in our Fashion for Good competition. And it was just a phone call. We skipped out the competition before that, which was like 150 companies applying to be in the competition. So we got straight in, and effectively there wasn't really a winner, but we're one of the few, like 3 companies that they actually ended up giving money to. So we kind of really got a big win out of that. Starting on April 21st, this process, we've been given a small bit of the V&A Fashion from Nature exhibition that starts on the 21st of April. So if you're in London, go down and check it out. You'll see some of the crazy things that we built in the lab and did in the lab just to put into the exhibit. And I think it'll be interesting otherwise, so don't just go for us, but I think it'll be pretty interesting. And I just wanted to bring back the fact that when we started out the project with arsenic, it really mattered to go to talk to people, government officials, and even the people, and particularly actually the people in the villages that were going to be end users of what the technology we're developing. Like the Ancient Mariner, our penance for developing new technologies is to talk to people about that. Okay, and I think there's a real lesson there. And so talking to people matters. And so thanks for your attention.
And that was, I think, really fascinating. Everything from what's happening at the moment with the industry and the harms there. I thought that image from China was terrifying.
Yeah.
To the amazing things that can be done. That we can do things that couldn't be done before, as well as being cleaner, cheaper, better in some ways. I think there's lots of aspects of that, about how we use bioengineering, the sort of plug-and-play ideas that we can do all sorts of things now. I have lots of things I'd love to ask about, but we have an opportunity for all of you to ask about anything that you have. We do have a microphone, and so if you could wait for it to come to you, there's one at the back there to start with.
And do remember to hold it like this.
Hi, I'm Kay. Thanks very much for all of your efforts and pioneering ways, particularly in this country, for dying, and hoping that you can take that internationally as well. I was wondering if you've heard of a fellow named Michael Brongort, who's a biochemist out of Germany.
Okay.
And he's also been working in a similar area, working with reinventing how to make various products. But one was one of their most notable works was a carpet that was a carpet manufacturer in Switzerland. Are you familiar with that one?
Yeah, I know of that. Yeah, I think that's really interesting. I think one of the things that is really important in this space, though, is that that people understand the regulation and understand what it is that's going into the product, so they go in with their eyes open. Because some of the things you could potentially make, for example, for dyes aren't particularly nice compounds in and of themselves. So we're trying to work towards things that will be fairly inert. You also have things that will, you have regulation that will tell you what you can and cannot make, and they will test it for those things, okay? So I don't know how they did that, but carpet is one of the great things to work with because it is, kind of the lowest level of fabric stuff,
Right, yeah.
okay? And so it's something that we would try to get into eventually as well, but yeah.
Yeah, I think there was probably a bit of similarities in your stories, and that's why I was wondering.
Yeah, no, I'm sure that, people that have looked at this, in fact, I was sort of surprised, 'cause, we've got, a patent on the process. And I was sort of shocked, actually, when we started doing the due diligence, how empty the space was. People just hadn't thought about this much, even though it's fairly obvious.
Thank you so much. Really interesting. And it all seems too good to be true. No, I mean, it really is.
Yeah, of course. Okay, so...
My only question would be, is this a very... No, I mean, I really think it is good, but is this a very high-tech industry compared to traditional dyeing industries, which you might find in developing countries? In other words, is this going to transfer the industry from poorer countries to richer countries who can afford the science, or not?
No, not really. In fact, one of the things that you worry about from an economic model is that anybody that has the bacteria can do it. Okay, so someone could just steal the bacteria from us and start up a dye shop anywhere. Okay, so so long as you have sugar to grow the things, you can do it anywhere.
Does it need a very high? Does it need a very sort of high amount of skill? A high level of science. That's my.
No, the science itself is actually quite low level. Okay, all you have to do is be a good microbiologist. So if you know how to brew beer, you can probably do this. All right. Yeah, it's not high tech at all. And in fact, what we're trying to do is partner with some dye houses in Italy so we can slot this directly into their current machinery. So it wouldn't change much.
One over there, and then at the back.
I am. Is there anything you're doing to try and stop people from being able to take your bacteria and then just sort of ferment it anywhere and sort of lose control?
Yes and no. I mean, I think we're starting to do things like barcode the bacteria in the genome, so we know if someone's using our stuff. But you can't exactly stop people from, because this is, once the designs are infinite, so you can design anything you want, and it'll still probably work. It's just that what you begin to realize from the fashion industry is that they're really, really particular about quality control. So they're not going to buy stuff from just anybody. It's a closed shop, okay? So part of the reason for going with the fashion industry is that they are like that, but also fashion can absorb the costs that we would incur greater than current practice now, although once to scale, it probably will go below their costs, simply because we can just keep growing stuff.
There's a question at the back.
Hi. What's the color fastness of the dyes? Do they last for very long?
Yeah, okay, so there's a couple, we haven't really explored the entire space, so we don't know, okay? But from what we've worked with so far, when you start looking, this is really weird, but when you start looking into what people do to dye fabric, they often have to use what's called a mordant, okay? That's why they use chromium, because they need something that will help the dye itself stick to the fabric, so it'll have better light fastness, rub fastness, and washed fastness, okay? So we're pretty average when it comes to light fastness. We're good when it comes to rub fastness, and we're good for when it comes to washing, because we can dry clean too, and it still seems to be okay. So what you begin to realize is that the bacterial cell itself holds a lot of mordants, okay? What you need are ions that have 2 positive charges on them. So we have lots of magnesium, calcium, and stuff in cells, because those are normal cellular components. Every cell has those things. So they just concentrate them out of the water in the cell. Well, guess what? That's really good if you want to attach what you're making as dye onto the fabric, okay? So the cells that's one of the reasons why they help attach the dye to the cell or to the fabric, and that's why they have these kinds of fastness properties.
Thank you.
All right, can I just ask about what happens in a landfill in normal fabrics with the chromium that's highly toxic?
Yeah, they just sit there in the
That just
landfill.
goes
Yeah, it leaches back
ground.
into the ground,
Or runs off into
runs
the
off or whatever, yeah.
management.
But I think,
So with your fabrics, are you looking at what happens in landfill? then?
yeah, so I think when you start, the whole fabric industry itself is going through sort of a revolution, because cotton is not a very good fabric or a good material, because the water used to grow cotton is massive, okay? And as a consequence, it's not the most eco-friendly thing. So there's other companies that are trying to make take cellulose from, like, I don't know, poplar trees or something that grows fast, and make them into, monomers of cellulose, and then make the polymer. And those things are probably more friendly. And we could go into, lots of things about fabric itself.
So we work a lot in India with factories and mills there. How do we persuade them to use your dyes? Because the biggest problem is getting it into the kind of right at the source. And yeah.
Yeah, so we've had conversations with people in India, and I think getting it into the factory, there's two hurdles. One is that people are very resistant to trying to retool, okay? So part of what we're doing here is trying to figure out ways that we can slot our process directly into the machinery that's currently being used. The other thing is that wherever they are, they're going to have to get GM clearance, okay? And depending on where you live, if that's easier or harder to do. And like with our arsenic biosensor work, I have kind of a strong ethical and moral stance on this. One of the reasons why we haven't proceeded with that project is because we couldn't get clearance from the EU to use a GM organism in the open, okay? And that's a problem. Factories are not so bad because they're like labs, they're closed environments, so it is much easier to get, you know, real clearance to do that. But so we're not doing anything until we can get, you know, the appropriate GM regulation cleared with what we do. Okay, and that's part of the reason we haven't really gone straight into places like India and China, because it's much more, well, it's unknown to me. Like, if it's here, I'm the GM officer for, you know, my department, so I know I can call up the guys at the, you know, HSE and say, we need to do this, and they're happy to work with us because they know us, but that's not going to work in China or India, okay? So what we're trying to do is to do it right, okay?
Hi. Have you done any work with textile printing? Because I know that's particularly polluting.
Yeah, I mean, so we just wanted to, did want to see whether our process could be, you know, made into a fairly fine thing. Because we want to try inkjet printing and that sort of thing, because you can do that with cells. And it kind of works, you know, so, you know, I don't know. I mean, I think the answer is I don't know at this point. But yeah, it's something we're certainly looking into. Yeah.
What are the challenges that you face in getting your bacteria to produce different pigment colours?
Well, I think the biggest challenge is going to be trying to make sure that whatever you take from another organism, you can take that pathway in a reasonable way and not suck down all the resources out of the cell. To, you know, because a lot of these things, so the one that we made here that's purple is based on the amino acid tryptophan. And so, you know, you can make lots of purple, but you're also sucking all the tryptophan out of the cell, and so they don't grow very well. So there's things where we're going to have to retool the rest of the cell to be able to redirect the things that they normally need or make towards that production. Okay, and those will be big challenges, I think.
Well, wait for any others. Can I just ask about the timescale?
Yeah.
When do you think I'll be able to go and buy something that's been dyed in this way?
Well, I don't know if you can buy it, but I can give you my cufflinks here. The answer is I don't know, because, again, this process is regulated. Okay, so if you have a new dye, that has to go through a regulatory regime that tells you that it's safe before you can actually release it for sale. So that takes some time, because they do a lot of different kinds of tests. You basically have to dye giant pieces of fabric. You send them to the test center, and they do all sorts of things to it to see how safe they think it is. Okay, so.
But years?
No. Probably, you know, year or so.
There's one over there and then one up here. Yeah.
Hello, could I ask about mordants, and is this a mammoth problem at the moment, or is it just a little problem? And could I also ask about the fashion industry?
Yeah, there's lots of things about mordants, okay? Mordants, basically, if you're a chemist, they're polyvalent cations for the most part, okay? So they're things that 2 positive charges or more to help the dye stick to the fabric. And it is a problem in the sense that most dyes that are not reactive dyes require a mordant of some sort to help it stick, okay? And
what we're all wearing at the moment in this room.
yeah, well, I mean, okay, so
will
some
be
of the
using
things, I don't know exactly the proportion, but I would guess that, you know, a lot of the really intensely colored things will be reactive dyes, where it's a chemical reaction between the fabric and the dye itself in order to stick. And that's why they're really light fast, they're very rub fast, they're very wash fast. but the chemicals that are involved in doing that process aren't very nice. The ones that are more traditional, basically, you kind of put the fabric into something that will let the fibers unravel a little bit, which is generally heat, which is why it costs a lot of money to do this. And then you put the dye in with a mordant, and it will help the dye stick to the fabric. Then it cools down, the fibers get rewrapped, and you wash out all the excess stuff, and you have a dyed product.
Thinking about the range of colours that are used and how frequently colour shifts in the end products.
Yeah.
How far across the spectrum do you think you'll be able to go in due course?
I think we'll probably be able to do almost any color, okay? We won't probably be able to do fluorescent things because the fluorescent things that we know about are proteins and they won't survive the process. But and making a true deep black will be hard because most organisms don't do that. But aside from that, I don't think there's going to be a lot of, what we're beginning to realize from the industry is that there will be dyes that will be banned. Okay, so there will be colors. Okay, like there's a red dye that's going to be banned like next year, and so a lot of places just won't make red stuff because you can't. So we're hoping that we can replace some of those things.
Thank you. In one of your penultimate slides, you had 3 flasks, the sugar solution, the density, and then the waste product. Is the waste product just a sugar solution, or what is the waste product that you're actually...
You're talking about this picture.
That one, yeah.
Yeah, it's basically a sugar solution with the transfer media. So basically it's got glycerin, sugar, and a few proprietary things in it that are, again, food grade sorts of things. So yeah, aside from the residual dye in there, if you put that and killed the bacteria in it, you could probably drink it, okay? The residual, well, it probably would give you diarrhea because I don't think you'd want to drink that much glycerol, but it's not a problem. So that's actually a good point because one of the things that we're trying to do is figure out how we can find another process that will use that spent dye liquor to do something else with it, because this is a continuing cycle that you need to deal with. You could just dump it in the river and it probably won't matter very much, but that's not what we want to do, okay? So we're looking into other microorganisms, things that will eat glycerol, and there are ones, and try to do something useful with those.
So you had a slide showing the sort of things you can use for the dyes, like the plants and what looked like a cochineal beetle.
Yeah.
Are you actually using the actual animals, or just synthesizing DNA from them?
Just taking the DNA. So most of these organisms, their genomes have been sequenced, so you can find out what the enzymes are pretty easily just by going on your computer. Right? This is a great thing about modern biology. So you don't need to do any of that. It's not like we have to go find the beetle or find the snail or any of that. We don't have to know that they exist. All we know is with their genome sequences, take that and re-engineer it and stick it in. So the answer is no.
Do you see a potential of applying this process to the actual manufacture of fabrics themselves rather than just the dyeing process?
There are people that are trying to do that. In fact, you can make cellulosic material from microbes, but you have to ask yourself, what is where are you getting your energy from, and what is the utility of that? Because there are companies that are taking, like I said, things like poplar trees that, they don't use a massive amount of water compared to other things. They grow fast enough that you can make cellulosic pulp out of them and retrieve that and then make it into fabric, okay? And those are probably energetically better things to do because you're getting your energy from the sun directly rather than feeding it sugar. So it's a better part of the process. But yeah, you can do that. But why? is sort of the question.
You guys are making me run.
So you were saying about how producing the dye for the bacteria can be costly. Is there potentially problems with like ejection of plastids that you're putting in, which would then mean that you've got a strain that doesn't have to produce the dye, so might have a competitive?
Yeah, there's always those sorts of issues. Like right now, we still have to use antibiotics to make the initial grow out, but we don't use them after that.
Right.
So yeah, so part of this is re-engineering the bacteria so they can handle the load that you're putting onto them. So there is a lot of strain engineering that goes on as well. So yeah, and we're redesigning the plasmids so that they're connected in a different way rather than antibiotic selection. So there's other pressures that will require them to be maintained in the cell.
Come on, somebody over there.
If there are no more questions, then, I mean, Jim, thank you very much for a really, really fascinating talk. I think one thing that I thought was really interesting, particularly given some of the views that are sometimes expressed about GMOs being bad for the environment. This is actually a really interesting example where use of these GMOs could be fantastically beneficial for the environment. So I think it's really interesting about how the science, the ideas, the fashion all comes together to do something, you know, that wouldn't sit in one single silo. So
no.
I found it really fascinating, and thank you very much, Jim.
Thanks. I hope it wasn't too long.
It was very good. More questions from people who've had many of the others as well.
Yeah.
I'll let you.
Yeah.
Hi, sorry, my name's Matthew Tennant.
Hi.
Sorry to interrupt. I'm Kay, and
Hi.
Nice to meet you.
See you, Julian.
Thanks for having me along.
No, it's fantastic.
And I just wanted to approach you about some of the other sustainability lectures that you have going on.
Okay.
This is the only one I've been able to attend this weekend, but do you have any others that are on the circular economy?
Not specifically, no. So our brief is to look at anything that's interesting and worthwhile, which means that we do everything. So we have done stuff on climate change reactions before. We might be doing some more on that. Not specifically circular economy.
Okay. Right. The reason I'm asking, a couple of reasons. One, I used to be involved with a sustainable enterprise initiative at Human Fiber Business School
Okay.
back in the US, being a professional.
Okay.
That was in the early 80s.
Oh, right, okay. What do you... And now you're...
Starting out like this now,
Okay.
but it's completely different.
Okay.
But I was...
Our volunteers all bailed at the last second.
That started that initiative, which then became the Centre for Sustainable Enterprise.
Okay.
Do you know if you're a speaker for the next session is around?
She's coming at 1 o'clock.
I was asked by them because I was
hang around
part of the startup. Not
Everything's set up across there.
Okay.
So there shouldn't be any issues if loads of that happens.
Okay.
And it sounds all right.
That's it.
So they're trying to show business
And that should be good to go.
there's different ways of thinking
Thank you.
about industries, different ways of thinking
Yeah.
about business. Here are example companies, and then I was sort of involved with the strategy
Okay.
developing the
Okay.
did my own MBA. Was a strategy tutor on an MBA program
Okay.
in UK.
Okay.
And did some case studies. This is back in 2007 for my
Okay.
students.
Yeah, there's a number of technologies out there that obviously done it.
So it's always been a passionate time. I've been involved both from industry
Good. If you give me a card, then I can.
I don't have any on me. I'm not having them reprinted, so that's lovely. I can give you what I did do. Write out a couple of reference books. So Stuart Hart, he was one of the academics that I was involved with. He's now a leader international. So if you can get him to come in and speak.
He's based where?
Don't know where he is now. He's Irish. He was head of architecture at University of Virginia when I met him.
Okay.
But he and Braungart have their own cradle-to-cradle international strategy and design consultancy. They work with China to do, those big cities.
Yeah.
So it's both the architecture
Yeah,
as well as the products. He might be money, but it is an academic thing.
We have just started.
And then they've got a book.
We have just started, so we moved into our facility in Norwich.
And a number of other things, but if you Google them, you'll find them.
Yeah.
My name.
How new we are. I guess
happy to have a coffee.
Yeah.
Give you some ideas.
So you're based where?
In Cambridge.
And whereabouts? I mean, do you say you have this?
Milton Village.
Okay.
Yeah.
And what's the new organisation doing?
Oh, sorry. Well, this is my company. Do you mean?
Yeah.
Well, there'll be an aspect of it that's research based, but not to worry. That's a much longer story, which I think we should probably talk about.
Okay.
That's like sometime. Sorry, that's a bit.
That's all right. No problem. Thank you very much.
Should I drop you an email?
Yeah, please do. That'd be really.
What would be helpful for you from planning your forums and your speaking?
So a lot of what I do is just chatting to people and finding out what's going on, what's interesting. So we have great facilities, as you can see. So it's trying to find things, the right mixture of interdisciplinary. So my brief is to get people thinking and talking, to do things that are interesting for fellow staff, students and alumni in particular. So the question is, what would be interesting for people here? So we're very flexible to different models. I mean, the finances aren't so important, we just balance it.
Yeah,
Right. Fantastic. Really good to see you.
Really nice to meet you.
All the best.